So, very good evening to you all. Yesterday we have learned about meiosis. This video is for class 10, cell cycle and cell division. We have learned how meiosis is important, what is its significance, why it is necessary in our body. Today we will be learning about chromosome, gene and DNA. Please listen to the video very carefully because chromosome, gene and DNA, these things are making quite confusion that how the things are interrelated to each other. Now, on being a depth study, when a depth study has been done, when we have opened the chromosome, we have found the chromosome is having stretches of DNA and histone proteins. On having a brief study of the chromosome, we found that chromosome is having stretches of DNA, means fragments of DNA, means the pieces of DNA as well as histone proteins. And this DNA is made up of gene. Like cell, no? We are saying the cell is the structural and functional unit of life. Similarly, the gene is the functional unit of the DNA. What is DNA? The genetic material, hereditary material. Full form, deoxyribonucleic acid. Now, when we are talking about DNA, or whenever we are talking about gene, or whenever we are talking about chromosome, in the chromosome, DNA is present. In the DNA, genes are present. So on breaking the chromosome, we will get DNA and histone protein. On breaking the DNA, we can get gene. So this is how it is interrelated. Now, having said this, we can see the chromosomes, how the chromosomes are formed. Chromosomes are formed during the time of cell division, and chromosomes are the condensed chromatin fibers. When the chromatin fibers are condensed during the time of cell division, they are forming the chromosomes. Now, having said this about chromosome, we can see the DNA also. We can see the gene also. Now, what is DNA? DNA is the genetic material which is transferred from our forefathers to us. And what is the gene? Gene is the functional unit of the DNA. Now, when we have studied in a very deep manner, we found that DNA is making a coil around the histone protein and thereby making the histone DNA and DNA and the histone protein together forms the nucleosome. Now, anyone is asking what is nucleosome? So, nucleosome is the combination of DNA and the histone protein. Means DNA is wrapped around the nucleosome to form the histone proteins. DNA is wrapped around the histones to form the nucleosome. Okay, so pardon for the previous statement. What I have said that DNA is wrapped around the histone protein. What I have said, the DNA is wrapped around the nucleosome. No, the DNA is wrapped around the histone. The DNA is wrapped around the histone protein so as to form the nucleosome. That is what the concept behind. When the DNA will be completely wrapped around. Now, when we are saying, is the DNA wrapped around nucleosome? Wrong. That's why I have made the previous statement, and that's why I asked pardon. That when the DNA is wrapped around the nucleosome, anyone is saying it is 100% wrong statement. No, DNA will never wrap around the nucleosome. When DNA will wrap around the histone protein, that time it will form the nucleosome. I guess it's clear. Now, wrapping of the DNA around the histone protein will form the nucleosome, and this nucleosome will compact and condense to form the chromatin fiber. And when the chromatin fiber will condense, it will form the chromosomes. So it is given in page number 12 in a very clear note that once the DNA is wrapped around the histone protein, it will form the nucleosome. Nucleosome will compact and condense to form the chromatin fiber. And during the time of cell division, I repeat my dears, during the time of cell division, what will happen? The chromatin fiber will condense to form the chromosome. And before the chromosome could go for the division, it will duplicate itself and it will form chromatids. So a very nice diagram is given on page number 13, figure 1.12. It is given that the centromere is in between. Now, there are few terms need to be discussed. What are centromere? Centromere are the point of connection between the two chromatids. And when the chromosome will duplicate itself, it will form sister chromatids also. When the chromosome will duplicate itself, it will form the sister chromatids also. Now, the joining point of the chromatids as well as the sister chromatid will be the same centromere. And the diagram which is shown in figure 1.12 is very much clear fact about it. In page number 13, figure 1.12. So what happens is when the nucleus will set to divide, at that time itself the chromatin fiber will condense to form the chromosome. Chromosomes on the either way will duplicate its chromatids, it will form sister chromatids, and all the arms of the chromatids, means total there will be 4 arms, means 2 pair of arms of the chromatid will be joining the same centromere. There will be no change. Now, what is the importance of chromosome? Why it is important? Yes, of course it is very much important because it contains the stretches of DNA, histone proteins, genes. It is a hereditary vehicle. Means it is carrying the hereditary material, that is the DNA as well as the gene, from our grandparents to our parents, from our parents to us. That is why it is important. It maintains the gender distance also. If the XX chromosome is there, that's a girl. If an XY chromosome is there, that is a boy. And that is the chromosome pattern only which decides the gender. That's why it is important. More than deciding of the gender, it is important to transfer the genetic material from forefathers to their children. And that is done by the chromosomes. No one else can do, only the chromosome can do it. Because chromosome is only consists of DNA and the genes. So structure of chromosome, if at all anyone will ask what is the structure of chromosome, how it look like. So chromosome will only form itself when there will be division of cell going on. And of course, our body is made up of more than 3.7 trillion cells. Approximately 3.7, we can't count, 3.7 trillion cells where the chromosome resides. Because 3.7 trillion cells means nucleus. And we cannot say that all the 3.7 trillion cell will divide at a time. Maybe some cell is still dividing in our body. Maybe some cell is taking rest. Maybe some cells are young. Maybe some cells are old and about to die. But certainly there is some kind of cell division 24 hours going in our body. That time the formation of chromosomes do takes place. And that time what happens? The sister chromatids will also form. Now what happens next? The sister chromatids, how it does form? When the chromosome duplicate itself, the sister chromatids will form. And 2 pairs of hands or 2 pairs of arms of the chromosomes will be having a close attachment with the centromere. So centromere is the point of attachment of the sister chromatids as well as the main chromatids, parental chromatids. I guess it's clear to you. The terminal end of the chromosome is known as the telomeres, the end of the chromosome. The end of the chromosome, the bottom part of the chromosome is known as the telomeres. Is that clear? Now, talking about gene. Gene is the hereditary unit which is transferred from our parents to ourselves and from ourselves to our children. So it's an hereditary unit. So whenever anyone is asking what is gene, so just we can say gene, either it is a functional unit of the DNA or directly we can say it's an hereditary unit. But if at all we are saying it's an hereditary vehicle, no, wrong. Because it is not carrying itself. It is being carried by the chromosomes. So if there is no chromosome, no passing of the gene. No passing of the gene. So for the passage of the genes, chromosomes are necessary. If there is no chromosome, there won't be any passing of the genes. Now, when we have gone through the gene and the DNA, we found DNA is not a simple structure. Rather, it is made up of nitrogenous bases. There are 4 nitrogenous bases in the DNA: adenine, thymine, guanine, and cytosine. A, T, G, C. A is having bond with T and G is having bond with C. A, T, G, C. Adenine, thymine, guanine, and cytosine. So this is the structure, how the DNA is being secreted in our body, how the DNA is formed in our body because of the presence of the nitrogenous bases. Now, today we have discussed till gene and I have just given the introduction of the DNA. Tomorrow we will be discussing more about DNA and we will know the main structure of the DNA and how the DNA is replicating. I guess it's clear to you. So the next video you will find the studies of the DNA, and as well please go through the video and match it with your text. Try to understand it.